This method can help answer key questions in the glycoprotein field, providing insights into function of the protein, the role of link glycans, and the mechanism of action of therapeutics targeting the glycoprotein.
The main advantage of this technique is that it can be applied to any glycoprotein of interest, facilitating the structural and biophysical characterization of a wide range of glycoprotein targets. Demonstrating this procedure will be Hong Kui, a research project coordinator in our laboratory.
To begin, prepare and transfect cells as outlined in the text protocol. Harvest the cells by centrifugation at 6,371 times G and 4 degrees Celsius for 20 minutes. Retain the supernatant, and then use a 0.22 micron filter to filter it. Load the filtered supernatant at 4 milliliters per minute onto a nickel NTA column in a benchtop chromatography system. After this, wash the column with 3 to 4 volumes of wash buffer. Elute the purified glycoprotein from the column using an elution buffer gradient while collecting fractions. Pool the fractions containing the eluted peak in a centrifugal filtration device with a 10 kilodalton nominal molecular weight limit. Then, concentrate by centrifuging at 4,000 times G and 4 degrees Celsius for 15 minutes, or until the sample reaches a volume of 500 microliters. Inject the concentrated glycoprotein into a 500 microliter sample loop. Load the glycoprotein onto a pre-equilibrated high-performance size exclusion column on an FPLC system at 4 degrees Celsius while collecting fractions. After this,